welcome to the cell membrane student notes. So I'll be going over these notes and I'll be writing extra things on the slide. I encourage you to do the same. Now just right off the bat, take a look at these three pictures. Would you be able to explain them to another student without reading a script? This picture that I zoomed into shows all of the embedded proteins. So go ahead and number these. I'm going to number them 1, 2, 3, 4, 5, 6, 7. There are 7 different types of proteins embedded into the cell membrane. In the following slides, we're going to go over each one of these and have a good grasp of what each of the proteins do for the cell. The cell is controlled by the nucleus in the sense of DNA makes RNA, which instructs how to make proteins, but it's the cell membrane that is the guard. Imagine modern day as a cell, all the gates, all the security guards, your student ID, passes from the dean's office. All of these are to allow students and faculty to move around and function properly at school. Well, the cell membrane has that same function. So we're going to go over these 7 different proteins. Your job is to take this picture without a script, hit record, just like I'm doing on my iPad, and record your voice over this picture, explaining each of the 7. If you can do that, you will have a very good understanding of the material. Don't be caught up just with writing an answer on the following slides. Don't just fill in the blank and then not really have a grasp of what the concept was. This presentation is filled with key terms that I will give you a Quizlet. So understand the term and the definition. You need to know the vocabulary to explain what you see in front of you. But don't just work on the Quizlet and vocabulary. Then you're not going to know how these proteins do their job, each one a little bit different. So your goal is to be a tutor and understand it so you can explain it to someone and explain it to yourself. It takes a little drill and practice, and that's what this presentation's about. So the first slide right here has some missing pieces for you to fill in, and we're going to get right to it. So fill in the blanks, but you're going to practice this so that you understand it. So there are a couple of names for the cell membrane. The cell membrane is also called the plasma membrane. Those are interchangeable words. The plasma membrane, it's built out of a component here called a phospholipid. It is a phospholipid. Circle the word lipid, and what does that mean? That means fat. So the cell membrane is made out of 2 layers of fat, usually drawn with straight lines like that. As I mentioned, I'm going to be doing a few other things on the slide. Engage with the presentation and add this to your slides as well. So this could be the inside of the cell. I'll say in. This could be the outside of the cell. Now inside the cell, it's 75% water. Outside the cell, it's usually 75% water. Water on the inside, water on the outside. So I circled a component of the fat. Notice that the tails are facing inward, and it's called the head is facing outward, either in or out. It's facing toward water. So this side likes water. I'll put loves water. And this side does not love water. This is where the fat is. So the tails are the fatty acid tails. Now, you've done dishes before, and you know that grease and oil, that's fat. It doesn't dissolve in water. They separate, they keep separated. So think about it. The cell is made out of a lot of water. It wants to contain itself. It's going to need a wrapper that will hold it in, and that is fat. So you understand the concept of 2 layers of fat wrapping up around the cell. So you can see it right there. So there's a lot of water in here, and there's water on the outside too. Do you recall water is oxygen and 2 hydrogens? This side is slightly positive. There's some electrons hanging out over here. So this side is slightly negative. Water is polar. Well, so are these heads here. These are polar as well. So water is polar. It's going to stick to the polar. Like positives and negatives, they stick together. Well, so do ions. Ions are either positive or ions are either negative. And they're going to stick through here. We're going to see how some things are going to easily pass through the cell membrane, but if they get stuck to the outer surface, they can't go through. So right here, this is an example of how the cell membrane is going to allow some things to pass through it. If you're not an ion, you can slip right through. If you're small, you can slip right through. By that, I mean if you are like a small molecule, you can go through right here. If you're an ion, you're going to get stuck right here if you're a positive ion. If you're a negative ion, you're going to get stuck here and not be able to go through. So that's how the cell can selectively allow things through it. This word permeate means to go in or out. There are 7 embedded proteins, and they're going to perform 7 different functions. And we'll get to it right now. The number 1 through 7 on that first slide don't matter, so I'm just going to kind of number them as we go through here. The first thing is molecules that are too big to fit through here have to go through a channel. It's a wider gate. Imagine if we only had one gate at the main entrance as you're dropped off at the parking lot. If you're waiting for 100 students to go through one gate, there can be a backup. If the cell has to get things through quickly, it's going to just widen the door, like have 2 gates. So this is just a wider entry. Notice that there are 6 molecules up here in blue and 1 down here. So in nature, things spread out. And they're going to spread out so that they have more elbow room. Imagine if these were like billiards. They're going to knock into each other and find, hey, there's more space over there without running into anything. And so, zip, they're going to go through here and find themselves in an area where there's less. So after a while, this will balance out so that there's like 3 there and 3 there. This is called equilibrium. In nature, things spread out with no energy required until they're evenly distributed across that barrier. So that's the first one. It's called a channel protein. Write this in. This is passive. That's in contrast to active over here. So passive, look it up in the dictionary. It means you go with the flow, you don't have an opinion, you just let things happen. So no energy required. It's kind of like sitting in an inner tube and just floating down the river. Now look at this channel protein that is called active. Now we're going from where there's one molecule to where there are 4 molecules. So normally it would go in the direction of where there's more to where there's less, the passive flow, but we want to go up the river. We want to go from where there's less to where there's more, and that will require energy in the form of ATP. So it's called active transport. There's another slide on this, so we'll get a little bit more detail. This is sometimes called an active protein pump because it has to pump in the opposite direction of normal flow. So this is still number 1 because it is a channel protein to widen the gap. So by the way, this is protein number 2 out of 7. The actual numbers don't matter, but we're going to count up to 7 of them. So here's the third one. It is called an attachment protein. So the protein is fully embedded into the membrane. And down here, it has an attachment to the microfilament that is inside. These are like ropes that are going to help to move things around inside the cell. So if you're going to help someone that is slipping down a hill, you're going to grab them with one arm. And if you aren't attached to a tree with your other arm, you're going to be pulled down with them. So this over here is the point of attachment for the microfilament. The other end of the microfilament will be attached to another object so that it can pull it toward itself. You'll also notice that the attachment point could be outside the cell, attached onto some microfilaments that are between cells, and so it provides an anchoring point. So you can see from the bullets that it can attach either on the outside or the inside. It can help support the membrane. It can help support the cell. And it can coordinate any changes in movement that have to happen. Moving on to number 4 protein. Again, all these proteins are embedded into the membrane. And these can move around left and right and back and forward. This is what it meant back here by being a fluid mosaic. If you've been upstairs by the counseling office, you will see a display case for Joseph de Hagers. He worked at Mater Dei for 50 years. He was an icon here. We have a day of celebration for him in the springtime. You'll learn more about him. Take a look at the picture in that display case upstairs. It's a picture of his face, but it's made out of tiny little boxes of student pictures. That's what a mosaic is. And it forms the picture of his face when you look a little bit farther back at hundreds of student pictures. That's what a mosaic is. Fluid means that things move around. All these things that are embedded, they are going to move around. We saw that clearly in the hidden life of the cell video. Things are constantly in motion. And that's what it means by a fluid. If you think about these guys as balloons in the ceiling, the entire ceiling's covered with red balloons and yellow ties. Grab one of those ties, walk across the room, and the balloons will bounce around and move. That's what it means by being fluid. The cell membrane is not a solid. It has things flow in a mosaic pattern. That's why it's called the fluid mosaic model. All right, so back to where we were. This molecule may have originated in your brain in a gland in your brain. Your brain controls your body, and to communicate, it has nerves, but it also can send chemical messages into the bloodstream. So imagine this guy traveling through the blood system, diffusing into this fluid that's in between your cells, and it's going to make its way to a receptor. You can see by the key and lock type of a fit, the shape has to match exactly the shape of the molecule. It will then trigger a response with the molecule that is held on the other side of the protein, and it might dislodge that so that it's available for the machinery of the cell. It might be needed in the Golgi apparatus or in the cytoplasm to start a chain of chemical reactions. So that is a signal that has to be turned on by a chemical. So that's cool how our cell membrane is going to receive a message from the outside and then make something happen on the inside of the cell. So that's what you see with these bullets down here. So you'll notice that I am paraphrasing. I'm talking with knowledge that I know. I've practiced this. This is your goal. You want to get to a point where you can explain things without just reading your script. You know, you can get through a presentation without knowing anything if you're just reading a teleprompter. If you want to get an A on any class presentations, have a lot of pictures, very few sentences, and you speak about the picture. Takes a little practice. Hey, if you're in theater, you're going to practice your lines. You might ad-lib a little bit. And so you could carry on the scene. So your goal is to understand the pictures and what's happening with these 7 proteins using the vocabulary, of course. So I'll give you a Quizlet and you can practice that. But don't stop there. You've got to practice your notes and understanding the pictures. If you want additional information, go into chapter 5 for this, and it is discussed in a couple of pages, the cell membrane. All right, that was number 4. Here's number 5, a junction protein. This is cell A and this is cell B. If there was not an attachment here, they might slide by each other or even pass each other by. But if they need to be tied together, there's a protein on this cell and there's a protein on that cell, and you can see by the curvature here, these proteins are 3-dimensional complex molecules that have shapes. Their molecules are going to combine together and hold them together. That's the junction. They're going to be tied together right there, and so they're not going to be separated. Did you know that Notability has this tape? So I just activated it, and over here on the notes, if this was your key term, now I taped over it. You could then go through your notes as a review, and now you're going to say what's underneath that tape. You tap on it, it is revealed. Tap again, and it's covered. Pretty handy tool. It's up here at the top, right there, right over here. That was number 5. Here's number 6. This is called a glycoprotein. Well, here's a prefix, glyco. That stands for glucose. Hopefully you're writing in a few extra things like I am. Don't just settle for the bare minimum, filling in the blanks. Make sure you understand everything on the slide. If you don't, put a question mark, put a star, say review, give yourself some notes throughout here and interact with them. So what is glucose? Is it a carbohydrate, fat, or protein? It is a carbohydrate. I'll say a carb for short. What are carbohydrates? They are sugar. So that's why it says attached sugar. Go ahead and circle this. I'm circling the protein, but that carbohydrate is attached to the protein. It's sticking up over the cell like an antenna or a cable dish, and it's just ready to communicate. Along comes cell B, if this is cell A, and it's got a perfect fit. Imagine that the glycoprotein is the hand and the glove is cell B. It has to fit right over that carbohydrate, and now they communicate. Think back to the hidden life of the cell video. The virus bounced on the surface of your cell, and it needed to attach onto that sugar, that glycoprotein, and say, hey, let me in. I've got a package here that you'd really like to have. It will help you. And it had the correct fit. And it was like a key that unlocked the glycoprotein, and it allowed it to enter into the cell. So it's kind of like your student ID tag. It marks you as you. So we have cells that come by and check, hey, how you doing? They hook up almost like a computer. In Star Wars, there's R2-D2 hooking into the computer and reading information. So that's what this point of attachment is. It is a communication center. So that was number 6. And I think we went over all of them. I'll review here at the end. So we've gone over this concept of active and passive on the first protein we talked about. And these are key terms that you really need to focus in on. Diffusion, it means spreading out. Sometimes these shapes don't quite work out, so I'll delete it. If you get your shape correctly, it will recognize, hey, you wanted an oval. And if you do an underline and hold it, it makes it a nice even. So diffusion is the movement of molecules when they're spreading out, when they're going from where there is more to where there's less, until it reaches equilibrium. So here is a phrase that describes that. Instead of saying more to less, you call it going down the concentration gradient. I'll draw like a ski hill right here. I'll put an up arrow right here, which means that this is where there is more. And then a down arrow right there, which means that this is where there is less. So now if I put an arrow here, this is going down the concentration gradient. From where there's more dots like there to where there's less dots right there. That is called going with the flow, or going down the concentration gradient. From the previous slide, this is passive. I'll change colors. If you're going from where there is less to where there is more, you are going up the concentration gradient. This requires energy in the form of ATP. It's kind of a shorthand. You'll hear this discussed as a concentration gradient, more and less on either ends. If you are going from where there's more to where there's less, this happens in nature all the time. It's diffusion. That's going down the concentration gradient. Go the opposite way. Grab your skis. There's no chairlift. There's no gondola. You have to hike up the hill. It's going to be tiresome. It's going to require you to burn energy, and your cells have to expend ATP in order to make it happen. So this is active down here. So now we've got some more information about being passive or active. The animation isn't working here on a static PDF file, but if you were to recognize that these are like ping pong balls dropped from a helicopter, and they're going to bounce around until they equal out. Sometimes there's a little bit more on one side or the other, but give it time and the forces of nature, and it will all be equal. It'll move around until it's equal, then keep bouncing around, but generally it will always be equal. This guy is like number 1. It is a channel protein, but it is allowing water to go through it. It's called aqua for water, and it says pour. If water has to pass through here in large quantities quickly, it will need the help of a channel protein. Water can occasionally get through here okay, because it's such a small molecule. But if 2,000 students need to get into campus, you need to have a wider gate. That's what this channel protein is. If it is carrying water, it's called an aquaporin. Now, the diffusion of water is so important that it gets its own name. It's called osmosis. Osmosis is the diffusion of water. So it's a key term that you need to know. All right, here's how active transport works. It requires energy, and it's going against the concentration gradient. You could also say up the concentration gradient. The energy required, it's ATP. So here we go. We are going to move molecules up the concentration gradient. How is it up? There's 3 down here, and there's 4 right there. Normally, things would go from the top down in diffusion as passive transport. But this is like an air dock or an airlock on the space station. So this is on the outside, and this is on the inside. And the astronaut is going to go into the chamber. It's closed to the outer space. Then it's going to close once they're inside. Going to close to the inside and open to the outside to allow it to go out. The energy required for this protein to change its shape, it's ATP. So that's why energy is required. And now you have 2 here and 6 up there. That's active transport. Occasionally the cell has to move things that would not fit through here. So what if there is a big blob right here, as big as I'm drawing it, that can't fit through the protein? So it's going to have to go right through the cell membrane. When large molecules have to go through the membrane, it is called exocytosis or endocytosis. Exo means out, endo means in. So on the next slide, we'll see this happen. Another name for a solid going through the cell membrane is phagocytosis, and the word cytosis is right there, cytoplasm. And we're going to see on the next slide that sometimes we have to have control. You don't want everything that bumps up against the membrane to be brought in, because that could be bad, right? Bring in a virus. No, you want to make sure that it has a chemical signal to enter. Just like when visitors come to Modern Day, they have to go over to the security gate and they have to check their ID and then they get a pass. So getting a pass allows you to pass through. Here's the picture. This is the inside of the cell, and it wants to get rid of this vesicle. This is the phospholipid bilayer that is going to fuse with the cell's membrane so that the contents get spilled out. That's exocytosis. Here we have endocytosis, bringing in large quantities of food. This is endocytosis. And here's the glob of food. It's too big to fit through a protein, so you can see the pseudopod is going to wrap itself around it to the point where it will be pinched off right there and become this membrane-enclosed vacuole. Here is the receptor. This is a glycoprotein. This is a carbohydrate attached to a protein embedded. And you have to have a signal, this purple triangle right there. That may have originated in another part of the body, like the brain. So the brain says, all right, it's okay to let this guy in. So floating in the bloodstream, heading over to the cells, this guy is going to attach onto the glycoprotein and allow the membrane to change shapes and wrap up around these green dots. It was these guys that we're trying to bring in. So now when it finishes the job, it is right here. It is a vesicle that is coated due to the response of connecting up with the receptor. So it's got the pass like, hey, I'm an allowed visitor inside of the campus of the cell. Let's wrap up these notes talking about energy. I'm sure in junior high you talked about the two forms of energy, kinetic energy and potential energy. Kinetic energy is energy of motion. Potential energy is stored, or it's the potential that it could be used for motion. Take a look at the analogy here. The potential energy is right over here. This is the fuel. Add this to your notes. This is the potential. This column. So gasoline is the potential energy in your car. As soon as it has a spark and oxygen, it is going to start a fire in the engine, and your engine will burn. Fire is burning the fuel. It gets up to like 300 degrees. That's why you have a radiator, to take some of that heat away from the engine. Never run your car without water in it, because 300 degrees will go up to 400 and 500, and it will melt the pistons into the framework of the engine, and you will ruin your engine. If you have an overheating car, turn it off immediately. Back to the concept. Potential energy. Gasoline is made out of carbon, carbon. So the number 87, 91, 93, this is a measure of how many carbons are in the molecule. The bigger the number, the more potential energy the gas has. So it's a little more expensive. But when it's combusted, it becomes energy conversion of kinetic. So this energy conversion column is the kinetic energy. Well, our mitochondria, it's the powerhouse of the cell. We're going to go over this in another class, but suffice to say that our potential energy is what we eat, and it's our blood glucose. This glucose gets into the cell, and it gets into the mitochondria. So does oxygen, and it will also not burn uncontrollably, but it is deconstructing the glucose, pulling the carbons out of this. Remember, glucose is carbon, 6 carbons, and the potential energy in glucose is going to be made into the kinetic energy of ATP and giving off heat too. So this is where we get our 98 degrees Fahrenheit. Just get the car got up to 300, we get some potential energy lost as heat. Your body temperature is coming from all of your mitochondria, the millions of mitochondria that you have. So that's what is called thermo. That's temperature. And it is some of the energy from the potential energy that's lost in the form of heat. They have a percentage for the amount of energy efficiency that our mitochondria are at, but I've forgotten that number. It's interesting. You could Google it. So our glucose is the energy of life. It goes from glucose, which is potential energy, to the kinetic energy of ATP and the heat generated. Okay, there are reactions that exo and endo. So this is in. Heat will go into the reaction, and this is heat going out. In other words, this guy gets hot, and this guy gets cold. So this is high potential energy. So the reactants have more energy stored in them than the product, so a lot of energy is given off as heat. That is an exogenotic reaction. Ones that will require heat to go in, this is going to make the product cold because the energy the reactants didn't have much energy, but the energy is going to be soaked into the products, and so it gets cold. So this is like a cold pack. I'm going to show that in class. You've seen these cold packs for emergencies, right? They're not cold until you shake them and get the reactants reacting together, and the heat from the reactants is going to go into the product and store more potential energy in the products, so the environment gets cold. And so that is something that we'll be talking about further. And the last topic here is about enzymes. Enzymes right here, they're a protein. Oh yeah, this is the 7th one. So this is the last one that is embedded into the cell membrane. It is going to attract two things to it so that they can react together, or it's going to take a big item and then break it apart. So it's either going to build up or break apart the reactants, either way. And so the terminology for these guys is the location in the protein where the substrate is going to fit is called the active site. The actual molecule, like the sugar, and in this case it was sucrose, this sucrose right here, that is called the substrate, and it fits into the active site. So those are terms. This is the protein. It is one that's going to speed up a chemical reaction without being used up. That's the definition of an enzyme. It's a protein that is going to help a chemical reaction happen fast. Without enzymes, sucrose right here might take a week to break apart. Here, snap your fingers and it happens in 1 second. Chemical reactions are sped up by enzymes. And how it speeds up the chemical reaction is that it decreases the energy required to get the reaction going, and it's a helper. So there you go. There's a rap song over at the notes. Check it out if you haven't seen it in class. It's brilliant. If you watch it, you'll see that this entire thing is made into a music video rap, and you can see everything happen. It's quite clever. And you should understand that rap video now. Take care, everybody. Use these notes and the Quizlet, but do not forget to practice your notes.